Good morning, everyone. This is our May EM-X, and this is the last of our current academic year, and we start again the first Monday of October. So I hope to see many people at the IMC, at the international meeting in Liverpool, which is my hometown, later in the year. And I'd like to welcome many participants who are logging in from Pico in Europe right now, and also from my Japanese companies class who might be here because we have our good friend Fujiyoshi-san from Japan, whom I've known for many years, who will be our first speaker, introduced by Wa, and followed by our friend Haimei at Berkeley, who has been very helpful, particularly for our students when our microscopes have been down. Jaime has really stepped up and helped our students get some good data. So I will say no more now, but I will pass the baton over to our colleague, Wachu, who will introduce, as is our custom, Dr. Fujiyoshi, who will be our first speaker today. Wa, over to you, please.
Okay, good morning, everyone. It's my great honor to introduce Professor Yoshinori Fujiyoshi from Institute of Science in Tokyo. I have known Yoshi for years. He was the pioneer in developing the liquid helium-electron microscope. And so he has multiple microscopes. When he set up the facility at Kyoto University, I personally spent a short sabbatical in his lab. It has been a wonderful host, incredible facility. And his facility was so great that he attracted many other international scientists from Europe and the States collecting very difficult data set from the liquid helium microscope, with which they were able to get atomic resolution even back in the early 90s. So because of his pioneering work, so he has been very influential to make the electron crystallography possible for membrane protein, which could not be solved otherwise. And then he will give us a survey of what he has been able to do. And in the last few years, he turned into doing cryo-EM for, again, on membrane proteins, which he target for designing drugs of various types to different targets. So he definitely have the respect of all our cryo-EM community as a pioneer to make the impossible possible. And then he is a member of the Japan Academy and many other honors as well. So it's my great honor to present Professor Fujiyoshi for our first speaker this morning. Yoshi.
Thank you very much for kind introduction. And now I try to show my slide. Could you see?
Yes.
Yes. Thank you very much for that kind introduction. First of all, I would like to express my sincere thanks to organizers, especially Wachu. He kindly introduced and Bob Sinclair, who is also my old friend. So today, I would like to discuss about our research result in my talk entitled Structural Physiology based on cryo-electron microscopy. It looks like no meaning, but I'm personally interested in molecular mechanisms, how education and other experiences in human development influence cognitive ability and personality in the adult. On parallel with such personal curiosity, studying personal curiosity, we are developing basic technologies for drug development as well introduced. For the aim, structure analysis of membrane protein is important, and I believe electron microscopy is a good candidate for structure analysis of membrane proteins. I believe the electron microscopy is suitable for thin observation of thin specimens such as membrane. However, in 1970s, electron microscopists were debating whether electron microscopy could produce an atomic image. Then I tried to confirm whether electron microscopy can enable us to take a molecular image of chlorinated kappa phthalocyanine. And here you will see the kappa atom, porphyrin, and 16 chlorine atoms are clearly resolved. By developing minimum dose system, we can easily adjust a best focus image of radiation-sensitive organic molecules such as silver TCNQ complex. However, radiation damage of biological sample is extremely serious, and after many struggling trials and despite not being able to obtain quantitatively accurate results, radiation damage is at very low temperature is significantly reduced compared with radiation damage at room temperature and liquid nitrogen temperature. Then we set out to develop a cryo-electron microscope in 1983 and perfected the superfluid helium stage to be built into conventional transmission electron microscope in 1986. From then, we made quite a bit of effort to improve resolution as well as operational difficulties of cryo-electron microscope. It is my great pleasure to honor for have a chance to explain our pioneers in structure analysis of membrane proteins, Richard Henderson and Nigel Unwin. They analyzed for the first time bacterial rhodopsin structure almost 50 years ago. Ion channels are the key molecule on signal transduction in nervous system, but most abundant element is water molecule. For securing ion channel functions, water channel has to exclude any ion permeation. 13 types of water channels were identified in our body. These channels are involved in numerous physiological processes. Intensive studies of water channels gave us puzzling questions. For example, aquaporin 1 permeates 3 billion water molecules per second and exclude any ion, even protons. The pH regulation for biological cell is crucially important because pH is strongly related with signal transduction, proliferation, and survival of cells. But hydronium ion does not need to move from one side to the other for changing pH condition. Proton easily pass through by changing hydrogen bonding partner of water wire as so-called Gross's mechanism. Therefore, proton exclusion requires separation of hydrogen bond that causes large potential barrier. Then water cannot pass through by such a high speed of 3 billion water molecules per second. Our structure analysis of aquaporin 1 revealed unusual fold of aquaporin from almost perfectly conserved NPA motif, asparagine fluorine alanine motif. The loop B and loop E forms a short helices. The asparagine of NPA motif back bonded with NH group of main chain by carbonyl groups. And as a result, 2 amino groups come inside the channel almost parallel to the channel axis. At NPA motifs, channel forms narrow constriction of 3 angstrom. Water molecule is 2.8 angstrom in diameter, and just 1 water molecule can pass through, whereas any hydrated ion and solutes cannot be permeated. Electrostatic field formed by 2 short helices forced to orient water oxygen of water molecule come close to the NPA center, and then oxygen of water molecule can smoothly form hydrogen bond with amino groups of 2 asparagine residues. The formation of these hydrogen bonds in turn orient 2 hydrogen atoms of water molecule in the pore constriction perpendicular to the channel axis. The orientation might thus be preventing from forming hydrogen bond with other water molecules because their arrangement perpendicular to the channel axis renders them too far away from adjacent water molecule. This is, we proposed hydrogen bond isolation mechanism. However, we could not observe any water molecule in the channel, and the water arrangement shown here is just a cartoon. Then, because we analyzed the structure is only 3.8 angstrom, high-resolution structure analysis of fast water permeation channel is required in confirm hydrogen bond isolation mechanism. Aquaporin 4 is a predominant water channel in brain expressed in endophyte of astrocyte and ependymal cells and glial lamella of hypothalamus. The water permeation speed of aquaporin 4 is identical with that of fast water permeation channel aquaporin 1. At 2.8 angstrom resolution, we could discriminate 8 water molecules in the channel. The densities of water molecule in the channel at higher resolution of 1.8 angstrom by X-ray were, however, blurred. This counterintuitive notion could be attributed to the difference of surrounding atmosphere of structure analysis achieved in lipid bilayer or not. We could actually analyze the structure of lipid molecule together with aquaporin 4 by electron crystallography. Lipid bilayer is known to give the characteristic dielectric constant. Characteristic distribution of the dielectric constant in membrane produces large dipole moment of these short helices, whereas dipole moment without lipid bilayer is very small, as theoretically shown by Sengupta et al. In correlation with electrostatic field formed by 2 short helices, the arrangement of the carbonyl group of channel acts as a binding site, highly hydrophobic surface, and lower the energy barrier for water molecule entering the narrow channel. These carbonyl groups might be essential for very fast water permeation through otherwise perfectly hydrophobic channel. These water-stable positions guided by polypeptide carbonyls and amide groups may make 3 billion or more water molecules pass through in a second. The distances between water position close to the NPA center are 3.9 angstrom, 3.8 angstrom, by which no hydrogen bond could be formed and proton permeation is inhibited. This is the hydrogen bond isolation mechanism I proposed. The transcellular channel with large pore is gap junction channel, which allows, of course, water and solutes and ions to pass through together. The gap junction channel have essential role in many important processes, including cardiac development, fertility immune system electric signaling. Gap junction channel can regulate ion permeation rapidly and permeate large solutes such as metabolites, nucleotides, peptides. Shoji Maeda analyzed the structure of connection by X-ray crystallography. Connection structure shows large channel constriction such as 14 angstrom in diameter. As you can see here, the crayfish can take nimbly flight. How such large channel can close and open so quickly. We try to analyze structure of innexin. We could analyze only poor resolution of innexins. Then we try to analyze innexin structure using single particle method. We could, and innexin structure showed a large channel constriction such as 18 angstrom in diameter. Our structure strongly suggests these densities could be assigned to lipid bilayer and also N-terminal part which might be pushed up by lipid bilayer. We could discriminate, not faint, it's faint, but an acyl chain of some lipid molecules. Therefore, we proposed closure of gap junction is mediated by phospholipid coming close into the pore, which resulted in the conformational change of the N-terminal from funnel position in open conformation to upper position in closed conformation. These structure analysis suggests attractive gating model of gap junction channel lipid-mediated gating mechanism. This model explains how channel can open and close very quickly, even for the channel as large as 18 angstrom in diameter. Now, let's go back to the water permeation channel. Aquaporin 2 is the predominant water channel in kidney collecting duct. When the body becomes dehydrated, when we feel thirsty, vasopressin stimulated phosphorylation of the C-terminal of aquaporin 2 stimulates it membrane transport from intracellular vesicle to cell surface, leading to the absorption of water from the primarily urine back into the body inside. In addition, nephrogenic diabetic insipidus occurs when the aquaporin 2 transport is abnormal due to mutation in aquaporin 2 genes. We then attempted to analyze structure of aquaporin 2. The aquaporin molecule, which is almost perfectly embedded in lipid bilayer, was previously impossible to analyze structure using single particle method. We actually tried to analyze aquaporin 2 structure by single particle method, but failed for many years. Use time is 3 or more years. We then developed 8th generation cryo-electron microscope. Using 8th generation cryo-electron microscope, we could eventually analyze structure of aquaporin 2. We have now analyzed many water channel structure by single particle cryo-electron microscopy. Water channels were classified into 3 groups: aquaporin, aquaglyceroporin, and orthodox aquaporins. Aquaglyceroporins are water channel that not only permeate water, but also small solutes such as glycerols. As no structural information was obtained, we studied aquaporin structure and function. The aquaporin structure analysis revealed that tyrosine 212 comes into the channel inside, close the channel, and come out and open yet. This type of gating structure is entirely novel among water channel family proteins. For understanding the characteristic loop E entering mechanism of aquaporin 3, we analyzed 3 structures: wild type and tyrosine 212 phenylalanine mutant and tyrosine 212 threonine mutant. And here you will see side view of monomer and extracellular view of monomer. And tyrosine 212 threonine mutant keep open and come out of tyrosine 212. And while the wild type tyrosine and phenylalanine come into and close structure. So these results suggest that aromatic residues is required. The 212 part of aquaporin 3 come into inside channel. We measured water permeability and glycerol permeability by stopped flow analysis. Tyrosine 212 threonine mutant shows fast water permeability of both. And these results are consistent with the observed structural feature. Tyrosine 212 threonine mutant has water permeability and channel keep open. Well, the unorthodox aquaporin 11 is an endoplasmic reticulum localized water channel essential for renal development. Its critical role in early renal development and function was revealed by knockout studies showing aquaporin 11 deficient mice develop fetal polycystic kidney shortly after birth. Despite this clear role of aquaporin 11, exact function of aquaporin 11 in transport remain controversial. Then we analyzed the structure of aquaporin 11 by single particle method. Notably, aquaporin 11 forms distinct homotrimer, which differ from tetrameric structure of all aquaporins. Structure comparison with orthodox aquaporin reveals that an additional N-terminal transmembrane helix renamed H0 play a critical role in this distinct trimeric assembly. Although this trimeric architecture lacks a rigid trimer interface and may be less stable, previously unreported disulfide bond between loop C and loop E on ER luminal side was revealed by stabilized unstable aquaporin 11 structure. Distinguishing feature of aquaporin 11 is its NPC motif, which replaced NPA motif that is essential for proton exclusion and water selectivity in traditional aquaporins, as I mentioned. The aquaporin 11 channel lumen is remarkably wide with a diameter at least forms even narrowest constriction. At arginine residue at ARR filter is replaced by arginine to leucine 219. Widened pore is shaped not only by unique composition of leucine, but also significant helix displacement, outward displacement of helix 2 by 4.5 angstrom with big shift and markedly expand pore radius. The cysteine 101 in NPC also expand the channel, which is not exhibiting aquaporin 4 and aquaporin 7 or NPA motifs. Such a broad channel architecture suggests aquaporin 11 is optimized as conduct large neutral solutes, notably hydrogen peroxide, H2O2, which plays a pivotal role in oxidative stress and intracellular signaling. So then I would like to shift the gear of my talk, and we developed mutation-tolerant inhibitor peptide against variable SARS-CoV-2 viral spike. Our strategy to develop a direct viral inhibitor that was no bad effect on humans. So although pharmaceutical companies have already developed drugs and vaccines, our peptide drug CeSPL works order of magnitude better than others. Importantly, has no side effect. And CeSPL's bronchial spray treatment reduced the viral load by 3 orders of magnitude compared to placebo. Pathological autopsy sections also demonstrated the efficacy of CeSPLs. We designed the peptide inhibitor CeSPLs based on structure-guided drug development. The viral RBD must bind to ACE2 angiotensin converting to enzyme 2 for infection to occur, and mutation on the binding surface are somehow restricted. Therefore, to maintain activity regardless of viral mutations, we designed the CeSPLs to bind to RBD surface similar to the ACE2 binding site. Because RBD surface of viral spike is almost flat and has no binding pocket for small chemicals, we designed the peptide drug. CeSPLs consist of 39 naturally occurring amino acids, forming helical bundle and salt bridge at the N and C termini. The main chain of RBD was confirmed to retain same structure as all mutant RBDs, and the part of CeSPLs shows purple have been designed to interact directly with main chain of RBD rather than side chain and are not affected by viral mutations. And our structural and functional analysis confirmed essential amino acid residues of the RBD for ACE2 binding. CeSPLs were then designed to interact with these critical residues and shown in green. Many mutations appeared on the RBD after Omicron BA1, but CeSPLs has strong activity against any mutant viruses. CeSPLs bind strongly to and dissociate less from all RBD mutant wild type to Omicron BA5 as shown surface plasmon resonance measurement. CeSPLs has high medical potency from 4 picomolar to 13 nanomolar IC50. Efficacy of CeSPS in vivo was verified by Syrian hamster. Administration of CeSPS led to increase in the weight of infected hamster. Viral RNA titer was reduced 3 order of magnitude, and total area of lesion part of lung section clearly shows the efficacy of effect of CeSPS. To the TOA assay means time of addition assay using CAL3 cell derived from human lung cell shows the potent activity of CeSPS against all variants, even by post-treatment. CeSPS inhibited in even subsequent round of the infection of human CAL3 cells. Large reduction in dead cell was confirmed by assay of Berkeley 6 cell compared with the condition without CeSPS. All cells died, but almost no CAL3 cell, Berkeley 6 cell died. The CeSPS works even severe case, preventing acute respiratory distress syndrome, ARDS. Even severe case of placebo treatment mice all died. CeSPS treated mice prevented from ARDS, and no mice died. To develop a drug candidate with medical activity against all types of influenza viruses, similar with mutation-tolerant inhibitor of CeSPS, we designed short inner peptide inhibitors for influenza viruses. Well, I have no time to discuss about GPCR, but we are interested in GPCR, which are a significant drug target. Before closing my talk, I would like to add a few words. We developed cryo-EM collaborating with JOL. Recent aquaporin structure was achieved by Shota Suzuki, Daisuke Kozai, Akiko Kamegawa. Previous work was nice collaboration with Thomas Baltz and Peter and Andreas Engel. The gap junction project was achieved definitely by Atsu Oshima, and CeSPS work first author is Shun Nakamura, and we have a nice collaboration with many researchers. I'm very happy to have such nice collaboration on this project. Thank you for having the patience to hear my poor English. Okay.
Wow, thank you very much for this very impressive volume of work in the last few years in particular. So if any questions from the audience, please post your question in the chat. While we are waiting for questions, maybe I can ask a first question. In the latest microscope you use to record all the single particle, what are the special components you have in the microscope? Do you have a liquid helium stage in that microscope? Or
yeah, and also cold field emission gun and omega filter and Gatan's K3 camera. These are very important.
I see. So in the liquid helium operations, how long can you run the microscope without refueling?
Well, I should mention for single particle analysis, we use liquid nitrogen temperature.
Oh, okay, that's what I meant. Yes.
Yeah, but helium, if we supply helium and 4 hours continuously use.
So before Bob, in the current microscope, can you run either in liquid nitrogen or liquid helium?
We are taking data liquid nitrogen. There are several reasons. Yeah, maybe you may already know very well, but when there are many difficulties to take single particle image at a very low temperature, for example, below 20 Kelvin, we confirmed these part is below 20 Kelvin, and gold particle can easily move around. This is quite old data, but we encounter this kind of trouble, and when try to take an image of single particle, we encounter difficulties of beam-induced particle movement. So we use liquid nitrogen temperature for single particle method.
Okay, good. Thank you. Bob, you have a question?
Yes, thanks, Hua. Oh, wonderful talk, Yoshi. Really, really impressive. Now, what I wanted to know was, you showed this mechanism of the proton movement from one water molecule to another. Now, can you, or when will we be able to see the hydrogen atoms in H2O? Or can you see the difference between H2O and H2O2, for instance, at all? Can you see the hydrogen atoms at liquid helium temperature?
No, we couldn't discriminate H2 and H2O2. So unfortunately, yeah. So we could see some densities at channel center, but yeah, we cannot discriminate.
Yeah, I think we've tried also, but yeah, it's difficult. When do you think we will be able to? Holy grail of electron microscopy.
Maybe, yeah. Someone will challenge. Yeah.
I think on the single particle, if we can get down close to 1 or better than 1 angstrom, we should be able to detect some sense of hydrogen.
Maybe, yes.
No, I think it's not maybe, it's definitely so. I mean, there are structure on single particle with close to 1 angstrom. So
yeah, but so far we can analyze water channels. These water channels are slightly better than 2 angstrom, 1.9 angstrom or so.
Right.
Now, so but we couldn't reach to 1 angstrom.
Right, but these are from 2D crystals, right? All the water.
No, single particle.
Oh, single particle. Okay.
Single particle analysis gave us slightly better resolution than 2 angstrom.
Okay. I want to point out about the liquid helium stage again. So we used your microscope years ago that you designed, then we were able to do the single particle cryo-EM, with which we can trace the backbone. So I think it may be worthwhile to revisit that using the single particle again, if you have time.
Chris Russo tried to improve technique to prepare, for example, small gold hole, and then Chris Russo, doing. But we are not so seriously doing.
Okay, good. All right, Kate, do you have a question?
Yeah, thank you, Dr. Fujiyoshi-san, for the great talk. I was curious when you showed the movement of the particles at liquid helium when you with the beam when you create those bubbles. Is there a way to mitigate for that, or if you went to lower dose?
Okay. Okay. I didn't explain. Can I explain? Yeah. So and I designed a gas introducing system, and this specimen stage cooled down to stage temperature is 4 Kelvin, and introduce nitrogen gas, and we can confirm solid nitrogen here. The bilateral ring shows a nitrogen ring, and then cover and eliminate bright illumination, make a hole, especially high stopping power means gold particle area temperature rise up. Water area is keep low temperature. Then bright illumination now expand and weaker density of. Then gradually cool down and disappear this hole.
I see.
Means solid nitrogen film is disappear means these part is below 20 Kelvin. Mm-hmm. So we confirmed electron beam illuminated area still below 20 Kelvin. And unfortunately, gold particle moving around heavily. I don't know why, but anyway, we encounter this kind of serious problem when we use liquid helium.
Interesting. So this was a liquid helium and gas flow stage that you designed.
So then, and also when we try to measure the radiation damage or cryoprotection factor, very complicated and not so reliable data we couldn't get. So we long years. I'm not sure helium stage cause problem, and we have to overcome anyway. But we couldn't, and So, but for if we try to use tomography or something, maybe radiation damage is very serious, and then we try to still develop helium stage and keep going.
Thank you.
Yeah.
Okay, there's one more question from Yap Brink. His question is: does your research allow any guess as to the structure of the ancestral aquaporin?
Pardon? Yeah, I'm sorry, I couldn't understand. Yeah. What?
Okay, so does your research allow you to deduce what are the ancestral structure of the aquaporin? Yeah, which one come first?
Which one come first?
Yeah.
Oh,
yeah.
I don't know. But this is a phylogenetic tree, and what do you think? I don't know.
Okay.
So there are many. Water channel is very life forms develop water channel at a very early stage. So even primitive life forms has water channel. Then many researchers studying this kind of phylogenetic tree of water channel. So, but I'm not specialist. So, sorry. So,
Okay, good. All right. Thank you very much, Yoshi, for these excellent
Thank you.
lectures. Okay, and
Thank you very much.
okay, Bob, it's all yours.
Right. Super. Thank you so much, Yoshi. And yeah, we're looking forward to trying your liquid helium stage sometime.
Yeah, and maybe we will develop a next generation. So.
Oh, excellent. Can't wait.
We'll try to. Yeah.
Thank you.
Okay, thank you.
So, and now our next speaker will be Dr. Haimei Zheng from Lawrence Berkeley National Lab, and she'll be introduced by her former student, Dr. Qi Zheng, not related, who is now with Professor Yi Che, myself, here at Stanford. So, Qi, please introduce your friend Haimei.
Yeah, thank you, Bob. Yeah, it's my great pleasure to introduce our next speaker, Professor Haimei Zheng. Haimei is a senior scientist in the Material Science Division at Lawrence Berkeley National Lab and an adjunct professor in the Department of Material Science and Engineering at UC Berkeley. Her research mainly focuses on nanomaterials, transformations, and dynamic phenomena at the solid-liquid interfaces, with a particular emphasis on the development and application of liquid phase transmission electron microscopy. She has received many honors, including the DOE Early Career, the Berkeley Lab Director Award, and the MIS Medal. She's also a fellow of both MIS, Materials Research Society, and MSA, Microscopy Society of America. So beyond this official introduction, a more personal one is the following. As Bob mentioned, I'm a previous PhD student in Haimei's group in the past 4 years. I still remember her very hands-on trainings, actually, teaching me how to fabricate the liquid cells, operating the microscope, aligning the microscope, designing research projects, and developing critical thinking in science. So no doubt, she's a very outstanding electron microscopist, material scientist, and a research mentor. So beyond in the lab, she's also very talented in the kitchen, actually, crafting signature cocktails and delicious Chinese dishes. I would assume Haimei possibly applies some magic nanoscale mechanisms in food science. Thank you, Haimei, for the mentorship and inspirations. So without any further ado, please join me in welcome Haimei for her talk, Development and Applications of Liquid Phase Transmission Electron Microscopy. Thank you.
Thanks, Qi, very much for the introduction, and thanks for the opportunity to present here. This is Haimei Zheng. I will be talking about development and applications of liquid phase TEM for material synthesis, energy materials dynamics, and more. Solution synthesis of materials, including many of types of syntheses. During the synthesis and reactions, structure, morphology, and chemical state of materials evolve in response to the chemical potential, external stimuli, and the nanoscale local environment. Nanomaterials transformations are intrinsically complex, and they often exhibit heterogeneity and fluctuations where defects, interfaces, intermediate states can strongly influence the transformation pathways. As a result, nanoscale transformations frequently deviate from thermodynamic predictions. These dynamic processes and their underlying mechanisms are often hard to probe using conventional approaches. Liquid phase electron microscopy can be a unique tool to study the dynamic processes. In my group, we have been developing and applying liquid phase electron microscopy as a primary platform to investigate a variety of physical-chemical processes at atomic level in real time by developing liquid cells, applying state-of-art imaging and image analysis method, and integrating computational modeling, we directly visualize dynamic materials transformation pathways and uncover mechanisms underlying nucleation, growth, self-assembly, dynamic phenomena at solid-liquid interfaces, and catalysis, and many processes. And this effort enable novel materials discovery and advance frontiers of energy research and related field. Liquid phase TEM can be traced to the very early days of electron microscopy development in 1944. The first liquid cell was made using a platinum supporting frame and a nitrocellular thin film from colloidal cotton. And water droplet was encapsulated and imaged, but the resolution was not better than optical microscope. In modern electron microscopy era, Francis Ross' team at IBM at that time developed liquid cells to study electrochemical deposition of copper, achieving 5 nanometer resolution. When I first entered this field, a major focus was developing liquid cells to improve resolution. I developed the much thinner liquid cell, achieving 1 nanometer resolution. The thinner liquid cell not only enabled high resolution, but also made chemical analysis using EDS possible, and it represent a significant breakthrough, enabled this direct observation of platinum nanoparticle growth. And we can revisit the previous movie, actually recorded on a videotape. In contrast, this is the recent movie recorded by my former postdoc, Tubo. This is a core shell nanoparticle growth, and it, in fact, nowadays we can look at much more complex reactions, and this shows the electroanalytical reaction, looking at the dynamics of the copper catalyst. And this is recorded by T, from a graduate student here. She recorded this carbon nitrogen onion structure. This is cuff material. And actually, these are very, very challenging experiment. And these advances represent remarkable progress in the field. Liquid phase electron microscopy has advanced significantly over the years. The field has been embraced by a growing international community, taking advantage of this platform and tackling diverse scientific challenges and achieving atomic resolution imaging through liquid and nowadays. However, achieving high resolution is only part of the challenge. Bridging liquid cell TEM observation with the real world chemical processes has been the central task. This requires careful control of reaction environment, mitigating electron beam effects, and ensuring what we observed within a tiny liquid cell reflect intrinsic the chemical reaction behavior in the chemistry lab, it demands significant effort and systematic and sustained effort looking into many different systems. This has been the motivation as well as the focus in my research group. The work in my group falls in these three categories of research. First, we tune the solution chemistry to enable more complex reactions, allowing us to look at many dynamic processes, including growth of many different nanostructures, and looking at different metals, metal oxides, and many different systems. And when these type of reactions very often conveniently triggered by electron beam irradiation. And we also investigate the extent by which electron beam induced reaction replicates the flask synthesis in the chemistry lab. And second, we use thermal heating to trigger the chemical reaction inside the liquid cell, very much similar to the conventional synthesis in the chemistry lab. The third, we develop electrochemical liquid cells, mostly to look at the beam systems, such as lithium-ion batteries, electrocatalysis, and over the years we look at many different systems. Before I show some examples of our study, I would like to emphasize the liquid cell development has been the very important for the success of our study and many of the progresses in this field. I highlight following the thinner liquid cell development, the graphene liquid cell, the thinnest membrane possible, further push the spatial resolution. Equally important are these putting functions in the liquid cell, including the thermal heating, electric biasing, and in fact, we only achieved very recently the atomic resolution using this polymer electrochemical liquid cell. Then I also wanted to emphasize the liquid phase electron microscopy benefit significantly from the recent advances in the detector development technology and imaging, advanced imaging method and imaging analysis, and key examples including the CMOS based detector, K2K3, and Datry, also nowadays, and electrostatic deflector, and techniques such as 4D STEM has been integrated into many experiments. Here, very briefly, and this is an example of our work in collaborating with Colin Opus when he was still in NCM, and Peter Ursis at the NCM of the Molecular Foundry. We use 4D STEM and to study the low temperature electrolyte, and using machine learning and the deep learning based analysis of 4D STEM datasets resolve the short-range ordering in this low temperature electrolyte. We found mechanisms similar to this high concentration electrolyte, and found this multiple, like 2 lithium ions. In the next, I'm going to show some examples of studying nucleation growth phase transformations, dynamics, and there's many different systems we have been studied. Earlier, we focused on the morphology changes and look at the rod formation, nanocube formation, 2D growth, and later we mostly change from this type of shape morphology growth to look at more complex structure formation, and especially we changed the growth from more looking into this kind of thermodynamic consideration to more non-equilibrium systems. And first, I'm going to show the example of 2D growth using 3D nanoparticles as intermediate. In this case, as you see, this is growth of cobalt oxide 2D nanosheet. We know many common 2D materials has layered structure in their bulk format. For example, graphene from the graphite, many variety of metal hydroxide. For these materials, this 2D growth is more reasonable from thermodynamic consideration. Mostly these lateral growth can be faster because of the lower energy. However, from the cobalt oxide, it has cubic structure. But the systems showing this 2D nanosheet growth is counterintuitive. We mostly look at this type of growth, and we found using this molecular precursor in the liquid cell, we formed this nucleation of nanoparticles first, and then they transform into 2D when they grow into certain size. And this is very interesting. We look at more detail and use a higher resolution, many different movies, and showing and they have this 2D type of growth. Again, very briefly, this is showing this, considering this thermodynamic approach. If we consider this textbook type of practical theory from 3D to 2D type of transformation, this is not energetically favorable because the volume is the same. Then increasing the this 2D growth, it would be introducing more energy. We collaborated with Dr. Lin Wang, and he established a very simple model using these conventional and volume energy, and including a shape factor, and really beautifully explained this type of phenomena in this plot showing what we observed very clearly. And when nanoparticles is small, and when the energy increases, when this 2D type of growth, L representing the spreading part of 2D, and when it is increase, this kind of L spreading, then energy increases. However, when a particle large enough, we can see the growth is spontaneous. However, later we found we have the opportunity to look at more, mostly with the increased the resolution. We look at much more details, and here is an example on the growth of core shell nanoparticle of cadmium chloride. And we again use molecular precursor, and we look at this adjacent particles. We found very interestingly, this type of growth as you see here, they form this 2D nanoplate. And we found this nanoparticle growing completely mediated by defects. What I'm going to show you the next is this big particle dissolve because it has an incomplete shell. This particle growing because this particle, the shell develop defects. And this is the next frame by frame showing the details, this comparing and this growth of this particle core growing because this incomplete, this locally no shell, and this particle does not grow until it develops defects. And this completely different from our conventional thinking of this Oswald ripening from this thermodynamic consideration. Actually, this shell or this core dissolve. It actually beginning. It has a bigger particle, and this is the growth of the nanoparticle. It developed this dislocation and nano cracks, and eventually all this mass come to this core through the nano crack. Then we are wondering whether this defect-mediated mechanisms is applicable to other systems. And we evaluate this through theoretical simulation. We collaborated with Professor David Limmer in chemistry department at UC Berkeley. He developed a very simplified model using just AB2 systems. This core shell allows this system fluctuate in response to the chemical potential. And it shows remarkably similar to what we observed when there is a crack developed in the shell, and then this core can grow very fast. However, it can stop growing when this crack is healed. And then we also found this crack is very much like considering this energy. However, we found these model systems remarkably reproduce what we observed, the basic kinetics observed in the experiment. This suggests that the mechanisms is general, likely occur in many other core shell nanoparticles. This work highlights the importance of tracking atomic pathways and cover the role of heterogeneity and defects in nanoscale materials transformation pathways. In the next, I'm going to show you the solid-liquid interfaces. In solid-liquid interface dynamics, these are intrinsically heterogeneous. They play a very important role in the wide range of chemical and electrochemical processes because they are buried in liquid. Many key phenomena, including the origin of heterogeneity and defects, the role of defects, intermediate transition state, are very hard to access. Using liquid phase TEM, and we can uniquely access this buried dynamic phenomena and address key challenges of the scientific problems. I'm going to show you some very exciting study we recently achieved. First is the inspired synthesis of high entropy alloy nanoparticles. And very briefly, the background. This is a very exciting, very hot topic. People have been studying high entropy alloy synthesis, and this plot showing the progress of the field from a few elements to many more elements together. And this not only for the fundamental science, but also this type of materials can be used for batteries, catalysis, and this type of synthesis either is conventionally from the high temperature lock-in, very much fast cooling, lock-in the high entropy state, or near equilibrium synthesis, solution synthesis. And these type of approaches have the drawbacks of the hard to control the morphology, or they are intrinsically applying to the thermodynamically miscible combinations. We created a new type of approach inspired by our liquid phase TEM observation. I'm going to show you this type of new strategy of synthesis, which is called isothermal solidification. And this is the liquid cell TEM experiment. We use the metal alloy here, and then we put in the ion solution of copper. We found interesting dynamics, fascinating. It has gas and it has solid liquid. Eventually, it solidified. Remarkably, we found the copper ions get inside this liquid metal. And this is very much inspiring. It gives us very important clue. And I'm introducing you this interesting new strategy we created using this phase diagram of copper, gallium, and this showing the conventional approach. Red arrow is the fast cooling of high entropy state. And the conventional approach, we created isothermal solidification, and this, like showing this nice cartoon picture, and these ions can be reduced at these interfaces of liquid metal, can be quickly incorporated into the liquid metal. And because this type of reduction and the salvation, the salvation involved in this liquid metal can be very fast. It almost no diffusion barrier. It can quickly introduce the oversaturation, getting into the system of supercooled state. And this can introduce the later solidification of this high entropy alloy, high entropy state. And this is a very fascinating new type of approach. We did many different calculations, and this we can calculate the reducibility, and we can estimate the miscibility, and we can create this map of showing different type of element, how easy to reduce, how easy to alloy, and it provides useful guidance for synthesis of alloys, high entropy alloys. And using this approach, we take advantage of this magical, this liquid metal and this ion interfaces, and taking advantages of this isothermal solidification, we can synthesize different type of morphology, control the crystallinity, and control the composition. It really demonstrates a new paradigm of synthesis, and this is a truly liquid TEM inspired synthesis. In the next, I will quickly show this development of electrochemical liquid cells for study of electrochemical processes related to electrocatalysis. And just comparison of this early study of this development study of lithium-ion battery, dendritic growth, and this type of silicon nitride liquid cell, we have this much thicker and the resolution is limited. And we developed this polymer liquid cell, much thinner liquid cell with digital electrode. We can achieve much better resolution and overcome many other issues common in this electrochemical liquid cell community. Here I show very briefly the development of polymer liquid cell, and we put a polyimide membrane on this insulated copper grade, aluminum oxide insulated copper grade, and we put interdigital electrode, and this is showing showing classical electrochemical behavior, and then we can use this type of liquid cell to have to study high resolution electrochemical processes, really challenging electrocatalysis processes I will show, and also it can compatible with cryo-EM, and we can fast freeze the intermediate state without shattering the membrane. And this is showing one example, very fast of this very quickly, and we found this copper this nanowire catalyst during CO2 reduction, and they're showing very interesting dynamics. We found this amorphous-like liquid, liquid-like amorphous state flowing at interfaces. It can be left and right, and it can also interconversion from the solid state and to the amorphous state. Because we have high resolution, we can find the detailed mode, how it flow, and how it interconversion. And this really representing the highest resolution dynamics in very delicate electrocatalysis complex system. As I mentioned, because we can do cryo-EM and fast like freeze this reaction state, we can look at this amorphous state very carefully, and this is showing this high resolution imaging of this copper state amorphous background, and we can do EDS use, and we can look at this copper with copper 1 plus and copper 0, and resolve this very challenging the puzzle in the field, and resolve this different composition and ratio. And we did many different control experiment to resolve the composition. I won't go through the detail. It includes these different this copper and also carbon, oxygen, hydrogen. It suggests this is amorphous is a copper complex. And we collaborated with a theorist, Dr. Zhigang Song, working with Professor Purana Narang at Harvard at that time, and Zhigang did a systematic study, and this is showing this kind of phonon calculation and really show this with electron doping, and the surface can be activated. And with this activation and this electron doped surface can be more active than without electron doping, and use MD simulation. He replicates this amorphous formation and showing this intermediate copper ion and copper zero dynamic transformation. And the next, I very briefly show that this platform of this electrochemical liquid cell allowing us to study many different systems, and this is copper, palladium, bimetallic catalyst, and we assemble them together, and we can study the dynamics. And we found even with this nanoparticle attached to the copper, during the biasing, those particles can detach, fly away. However, we made this Janus particle with very epitaxial interfaces between copper and palladium, and we can observe copper dissolve and deposit at the other side of these palladium nanoparticles, and showing those dynamics. And I also wanted to show we look at many different systems, and here is another example looking at a copper oxide pre-catalyst evolution during CO2 reduction. And during the reduction, this copper oxide quickly form this copper with many nanocracks, and this is a beautiful catalyst, activated catalyst, because the reactant and reaction product can come and go to and from the catalytic performance, the surface, and contributing to the enhanced catalytic performance. The very last, I wanted to show this system on the heterogeneous corrosion pathways of platinum nickel nanoparticles. I highlight this work because liquid TEM is often technically very demanding and challenging. We very much relying on experienced researchers like postdocs and advanced in their higher level senior graduate student, and this was done by our first year graduate student Haimei Zheng, and without any TM background before, and he completed this experiment mostly during the summer. And I just very quickly explain this synthesized nanoparticle, core shell nanoparticle with this nickel-rich core and platinum shell, and we were able to track this etching of the core and breaking of these later the frames. And this we can rationalize this observation using this type of intercalation theory, and reported in this work. The very last, I would like to acknowledge and thank my group members and collaborators and DOE-BS for funding, and thank you for your attention, and I will be happy to answer any questions.
Great talk, Haimei. Yeah, it's always great to see these fascinating movies. Yeah, just a quick reminder to the audience, feel free to input your questions in the Q&A panel, and we will ask for you. Yeah, I can start with the first one. So it's very amazing to see the high resolution TM like in situ movies. Can you talk a little bit more about the thickness of the liquid inside your liquid cell?
Yeah, the thickness can be controllable. Earlier we used the silicon nitride with controlling the spacer, mostly within 100 nanometers range. However, using this membrane liquid cell, we can achieve many type of thin pocket. We can measure the thickness of this pocket, and the thinner one we focused on more thinner, like a tenth of nanometer thickness of liquid.
Yeah, thank you. Bob, you have a question. Yeah.
Yeah, thanks. Thank you very much, Jaime. I think could you talk a little bit about the ionization effects of the electron beam on the liquid and how that might change the chemistry and then change the reactions compared to bulk, which you can't see?
Yeah,
typically, what dose and what dose rate are you using, and how does that compare with what Dr. Fujiyoshi has been using as well?
Very good question, Bob. Actually, this is a very important question for any system we study. We know this ionization and knock-on damage. And for systems, we track every individual system control, do the control experiment. For the electrocatalysis I show, and that system actually reasonably robust. The threshold is around 2,000 electrons per angstrom square per second. Then for the battery research, and it's actually very much comparable to the biological imaging, like 1 electron per angstrom square per second. But this system is the electron beam is often very complex. I didn't have time to show, and this actually very different system have different kind of threshold the electron beam dose. And this shows the earlier study of lead selenide. We can see that if we look at this in water, it dissolves. And we went to actually use your facility in Stanford and use flow oxygen. It also dissolves. And we look at with this kind of slightly this with open to the air, and we also see the dissolve. And however, we assemble this nanoparticle in a glove box and seal it. And we found without exposure to moisture, to oxygen, the tolerance dose in the liquid and can be much higher. And this goes to there's a 3,000 electron per angstrom per second. This very high electron dose threshold.
Yeah, we try and use as low a dose as possible, and even so, there's often effects even at a very low dose as well, in our experience. Thank you, Jaime and Chi. Back to you, please.
Yeah, Kat, go ahead. Yeah.
Yeah, thank you so much, Jaime. I had a similar question to Bob, but on your isothermal synthesis of the high entropy alloys. I really enjoyed that aspect. And yeah, I wanted to ask for that isothermal synthesis. Number one, is radiolysis. Do you think radiolysis is a large part of why you can synthesize these out of equilibrium structures? And then B, when you're going, say you're going, down in a phase diagram in temperature, you know what your composition is, and you're just, for that quenching of the high entropy alloys. But when you're doing this kind of new isothermal synthesis route, how do you determine what your composition, like where in the phase diagram you're going to end? Where in temperature, but where in composition? How do you determine that?
And thanks for the question. All the synthesis actually is outside of the microscope, and we actually have a patent that we can make a large amount completely outside the microscope. And this is just the inspiration. I show this kind of getting together inside of this liquid alloy. All the synthesis is outside of the microscope.
So is this a bubble here, that video? Is that a bubble that's being created by the beam, or you think that happens outside the microscope also?
This gallium reduction, and this at the interface, it generates hydrogen gases. And so it like a star bar is a natural reactor here, showing this is a bubble. There's from gallium reaction, but there's no beam effects in the outside of the microscope because we are showing large amounts. We can use this. We apply this high entropy alloy particle for catalysis and for coating and for many different applications. And this just is inspiration. And this also showing just the concept, controlling the composition. This just show this really diffusion. There's no diffusion barrier if it just reduced by single atom, and this get oversaturation quickly, getting into a supercooled state. And the composition can be controlled by the precursor concentration. We can vary the composition based on the precursor and also the liquid metal composition beginning with. In addition to pure gallium, we can also use the gallium indium tin as a precursor.
Got it. Thank you. Thanks so much.
Okay, Chi, I think we are out of our time fairly soon. So I think, do you have any more questions for Haimei, Chi?
Not for now. Yeah, I will return back to you, Bob and Hua. Yeah, to wrapping up the session.
Thank you.
Yeah,
Thank you
thank you,
so much, Haimei and Yoshi. But Hua, would you like to say a few words, please, and maybe Yoshi also, before we sign off?
Yeah, let's open for informal discussions, and you can stop the recording. Yoshi, I'd like to drill on the scope that you are developing. So right now you use is that the ARM300 you are using to collect the single particle data, or something completely different?
300 kilovolt. Yeah.
So I believe that Prasanna cannot unmute us. We're still being recorded, Hua.
That's okay. Those are normal questions.